Hello everyone, I Assistant Professor V Shete from Department of Microbiology, KJ Somaya College. In this online lecture, we will start about studying the rabies virus. Okay, so let's start. We will study first the structural characteristic and the genomic properties of its virus. So the structure of the rabies virus is having the bullet shaped structure. As you can see in the right hand side picture, the shape of the virus is the bullet shaped virus, right? And it is having the 180 into 75 nanometer in the size, with one end is rounded. Okay, you can see in the topmost end, that is the rounded end, and the above the second one end is the planar or the concave end. Then it is the enveloped virus. Okay, and in that envelope, this envelope will contain the G protein and the M protein. Okay, now what is the G protein? These are the glycoprotein. You can see in that picture the blue color projections, that are spikes which are pointed outwards. Okay, these are the glycoprotein. These are composed of these glycoproteins, surface antigens. Okay, now this glycoproteins has different important roles, which are those: first, it will mediate the binding of the virus, okay, to the acetylcholine receptor in the neural tissues. Basically, what is the rabies? See, rabies disease is the inflammation of the brain, that is it will cause the encephalitis. Okay, so it is very much in contact with the CNS, the central nervous system, right? And therefore the G protein will help the virus to reach to that site, to the site of the infection. Now the properties of this each protein we will study in the later slides. Now first we'll come to the next protein, which is the M protein, called as the matrix protein, which is present beneath the envelope. Okay, then we have the genome. The genome is unsegmented, which is continuous, linear, unsegmented, single-stranded RNA, okay, which is negatively sense. Alright, this RNA will contain the RNA-dependent RNA polymerase. Okay, RNA-dependent RNA polymerase enzyme. It will contain. Okay, you can see in that picture the genome is shown. Also, you can see the black color genome has been shown. So that black color is the outline. Okay, and inside that there is the RNA strand is present. The black color is the capsid. It is the nucleocapsid. It is the coat of that genomic material, the nucleic acid. All right, and that capsid will contain the RNA dependent RNA polymerase and also other some structural genes or the antigens, okay, proteins. This is the structure and the genomic properties of the viruses. Now, which family this virus belongs to? The family called as the rhabdoviruses. Okay, rhabdos means rod, and that's why the name is called as the rhabdoviruses. Okay, and the genus, now this family is also divided into the two genera. First is the vesiculovirus, okay, and vesiculovirus, and the second one is the Lisa virus. Now this Lisa virus is coming under this, our rabies virus will come into the second genus, that is the Lisa virus, and in the Lisa virus also type 1, Lisa virus type 1. Okay, why Lisa? Lisa means the rage. Rage is the synonym for the rabies. Okay, this is the structure and the genome of the rabies virus. Now we will see the resistance properties of the rabies. Now the rabies virus is resistant to the sensitive to the ethanol, iodine, then soap or the detergents, okay, then a quaternary ammonium compounds, lipid solvent, okay, lipid solvent, that is the ethanol and the chloroform and the acetone, okay. Then it can also inactivated by the phenols, formalin, beta propiolactone, okay. And this beta propiolactone is used for preparing the vaccines also, okay. We have the one vaccine that will come in the later part, that is called as the Bpl vaccine, the beta propiolactone vaccine, which is this chemical is used to inactivate that virus, okay. The vaccine is containing the 5% ship brain cells ship brain tissues, and that is containing viruses, right? So that virus are getting inactivated with the help of this Bpl, beta propiolactone, okay. Then it can also be destroyed at the 50 degrees Celsius for 1 hour and the heating at 60 degrees Celsius for 5 minutes, okay. And if we want to store this virus, then it can be preserved at the minus 70 degrees Celsius or with the help of the lyophilization. What is lyophilization? Is the storage at very low temperature, okay. Then if you want to store in the dry ice, the virus has to be sealed very carefully in the vials, okay. And also it can survive at the 4 degrees Celsius. Alright, so these are the resistance and the susceptible properties of this virus. It is deactivated by most of the chemicals, right? Okay. And if we want to preserve or if we want to store the virus, then we can store with the at the minus 70 degrees Celsius or with the help of the lyophilization process. And also we can store this virus and the dry ice, okay, and there the virus has to be sealed in the vial, alright. Then we will see the antigenic properties of this virus. Now as we have saw the antigens that are present on this structure, which are those antigen? G protein, M protein and some other glycoproteins, okay, other membrane proteins. Now starting with the glycoprotein, the G proteins, which is very important protein or antigen in the pathogenesis, which will mediate the viral genome to reach towards site of the infection, right. Now now I will tell the properties of that G protein, the glycoprotein, okay. Now first it will mediate the binding of the virus, right, binding of the virus to the neural tissues. Then second, it will induce the heme agglutination inhibition anti-activity, HI antibodies, okay. Now this rabies virus possesses the heme agglutinating activity optimum at minus 4 degrees Celsius and the 6.2 pH, okay. Now it can also induce the neutralizing antibodies, which are the protective antibodies, okay. And as it can induce the neutralizing antibodies, therefore this can also be used in the preparation for the vaccines, okay. Then it stimulates the cytotoxic T cell immunity, and as it will induce the cytotoxic T cell immunity, it will help the host cell to produce antibodies against that virus, okay. Then the purified glycoprotein may act as a safe and effective subunit vaccine, This is a type of a vaccine, subunit vaccine, that we will see in the later part. But this glycoproteins are very much important because this can be also used as the vaccine preparation, okay, for the vaccine, and which is very safe and the effective vaccine, right? See, it will, as it is inducing the immunity, then it is a protective antibodies. It will also induce the protective antibodies, right? And therefore, due to such properties, it can used in the vaccine preparation, okay? Then second antigen that is present is the nucleoprotein, okay? The nucleoprotein, why nucleoprotein? That is the nucleocapsid protein present in the nucleocapsid, okay. Now it will also has the certain properties. Now which are those properties? It induces the complement fixing antibodies, okay. It will induce the complement fixing antibodies. Then therefore it can be determined using the CFT, complement fixation test. Then the antibodies in this are not protective antibodies. That means this antibodies will disappear after certain duration of the time, whereas the protective antibodies remain in the host cell for very much longer durations. All right. Then the antigen is very group specific, and the cross reactions can happen with this antigen, okay, with the similar type of the virus or with the same species of this virus. Then the anti-serum prepared against the nucleocapsid antigen can be used in the diagnostic immunofluorescence test, okay. In the immunofluorescence test, we used to determine the antigen with the help of the monoclonal antibodies. That antibodies are attached or are incorporated with the fluorescent molecules, okay. And therefore if the antigen is present in the sample, then antigen will bind to that antibodies and the fluorescence will emit. This is the immunofluorescence techniques, right? Then this anti-sera that is prepared for this nucleoprotein antigen, that we can use to determine this antigens with the help of the immunofluorescence methods, okay. Now we'll come to the other proteins. What are the other antigens? Such as the two membrane proteins majorly. First is the RNA dependent RNA polymerase, which is responsible for the polymerization, okay. And the second one is the glycolipid. These two proteinic antigens that are present as the other proteins. See, RNA dependent RNA polymerase is also present in the genome, okay, in the RNA. And with this, the glycolipid is also present, okay. So in this lecture, we have studied the structure of the rabies virus, the genomic properties of the rabies virus. Then we have saw the resistance properties and the susceptibility properties of the rabies virus, and then we saw the antigenic properties of this rabies virus. Now I hope you understood this online lecture. If you have any doubt regarding this, please ask in the comment section. Thank you for watching.